We are really thrilled to be able to do this and to welcome all of you. I'm Michael Chaim. I do the development and some other things for the McPherson ERI Research Institute, and I've been in touch with many of you, not only over the weeks leading up to this, but before that. In a normal year, we would be having this event at the Weissman Center, as we've done in past years. That's where this update began. We started it as a thank you for people who had joined us for Cycle for Sight, which I think most of you know is our annual fundraising event that we do in March every year. And of course, getting together at the Weissman Center is not really an option for us this year, but the upside is that we do have people from all around the country, certainly all around Wisconsin, who are joining us for this and don't even have to worry about parking on campus. So that's another little plus there. You know, like many of you, I've been working from home for the last 3 or 4 months. So it's really just a pleasure to be able to see people, even if it's this way. And I look forward to getting back to meeting with people one-on-one or in small groups again. But since that will be some time probably, we're glad to be able to do this. And assuming that all goes well, I think we'll probably do it again, maybe before we get back together as well. I'm going to introduce Dave Gam, and then we're going to start. Dave has been the director of the McPherson ERI Research since 2012. He's a professor in the Department of Ophthalmology and Visual Sciences here at UW-Madison, and he's also the Sandra Lemke-Trout Chair in Eye Research. But I think many of you are familiar with the work that he's doing in the GAM lab, developing methods of photoreceptor replacement and regeneration, which we'll talk about today, along with some other things. So thank you, and I'm going to turn this over to Dave.
All right, thank you, Michael, and thanks to everybody. Again, great to see a lot of names I'm familiar with and friends, and some new names as well. I'm going to break my part into really 3 sections. As Michael mentioned, we really want to set aside a lot of time for question and answer. I'm willing to stay on and answer folk questions as need be, and that's what's kind of nice about it. But before we get to the question and answer, I thought it would be good to have a bit of a primer and let everyone know kind of from a broad perspective where we're at. And that's going to be broken down really into 2 sections. The first will be kind of a general overview, just establishing kind of what we do and the different types of diseases and the challenges that they bring forth. And then hit some highlights of some of the research that's being done since our last update. Obviously, we can't go through everything exhaustively, but I'm going to hit on a few. And as folks have questions, I'll discuss them. And my answers will not be confined to what's just going on at the McPherson ERI Research Institute. We think we're the best in the world at what we do, but there's a lot of other work being done around the world and a lot of folks that we collaborate with. So I will do my best to get you informed of what's going on out there. So the first part then is just to go over for everybody in the audience. And some of this is going to be very, very basic for some and maybe a little bit advanced for others, but hopefully it'll be helpful to put further discussions into context. So the first question I have is, where can things go wrong? When we say we lose vision or we are blind or otherwise have issues, what does that mean? Where are the targets for various types of disease? And so we can break this down kind of into 4 parts. We have the front of the eye, which is sort of the window into the visual system. It's important to let the light in. It's important to be able to focus light. And then we have the back of the eye, or the retina, which is like the film in the camera. So if you think of the eyeball itself like a camera, you have the lens of the camera in the front and you have the film in the back. And much like film, it's very thin, and it has a layer within it that detects the light, sort of like the silver grains, and then it traverses the signal back to the brain. And the pathway to take all of those signals, those millions upon millions and millions of signals that your eyeball is registering every single second in microsecond, is the optic nerve. So it consolidates that information and sends it back through 1.2 million little wires back to the visual centers of the brain. And what we're showing at the back of the brain is the primary visual cortex, but upwards of about half of the brain has some sort of visual input. So there's a lot of different brain injuries that can affect visual function in some way or another. So that's how your basic visual system is laid out, but what can go wrong? Well, just like any sort of pathway, any disruption along this entire length of the visual system can lead to visual disruption. So in the front of the eye, you can have things that we think of as relatively simple, like nearsightedness or farsightedness or astigmatism. things that we take for granted in the Western world that could just be corrected by glasses, but in a lot of folks around the world that don't have access to glasses, it can be a huge impediment to being able to work and to being able to function normally. There's also infections, opacities of the cornea, scarring of the cornea. The cornea is that clear dome that covers the pupil. And if that is opaque, then you can't see through that. And then we have cataracts. We all know about cataracts, which again, we can take care of here in the Western world, but in other parts of the world, we cannot. And then trauma. So those are the sort of things that can affect the front part of the eye or the window onto the visual system. In the back of the eye, we have, again, trauma can be anywhere along the visual system, of course, so we include that. You've probably heard of retinal detachments, where that film, which is supposed to be plastered up against the back of the eye, peels off, much like bad wallpaper. Inflammatory diseases can attack that film. Retinal strokes or bleeds. Diabetic retinopathy is very, very prevalent and growing in prevalence. Macular degeneration, retinitis pigmentosa, and other genetic diseases that can lead to an intrinsic degeneration of that delicate film that's responsible for detecting light. In the optic nerve, we have glaucoma, one of the most common causes of vision loss in the world. Inflammatory diseases like optic neuritis, which we'll see with multiple sclerosis and other diseases. And infections can occur there. Tumors can press on the optic nerve and squeeze it and cause it to not function normal. And again, trauma. And there are genetic diseases that cause the optic nerve not to function normally. And then in the brain, we can have head injury, birth damage, stroke, and tumors. So can we treat any of this effectively currently? And I would say in the front part of the eye, yes. So we have really wonderful cataract surgeons. We have the ability to take out cataracts and put artificial lenses in that are more and more sophisticated. We have the ability to take off the cornea and put a new cornea on, a so-called corneal transplant, that when you donate your eyes, that's really what you're doing. You're donating the cornea to help somebody see who has cloudiness or opacity of the cornea. A lot of trauma we can help or fix, and of course glasses like we talked about before. But as we go back more and more into the visual system, our ability to really affect the disease in a positive way kind of goes down. So when we talk about optic nerves, things like glaucoma, for the majority of people, yes, we have drops that can control pressure, and we have the ability to stave off the really bad parts of the disease. So most people who have glaucoma can be treated effectively by our glaucoma specialists, and they'll have minimal or no noticeable vision loss. But there are unfortunately people who fail all of the treatments that we have available to us today and progressively go blind. Same thing with inflammatory diseases. We have medications that can control that, steroid or immunomodulatory agents, but not everybody is effectively treated by those therapeutics. Same thing with all the other things we've mentioned here. When it comes to the retina, it's even a little more dire. Of course, retinal detachments, if they are not involving the central part of the retina, can be reattached, and we have great retinal surgeons who can do that. We have the ability to use laser and other approaches to control diabetic retinopathy, but oftentimes we're just putting our finger into a dam. And ultimately, if too many holes pop up, the dam will break. So we can stave things off, and we have some good treatments. But for many of these, like macular degeneration and retinitis pigmentosa, we have no treatments or very few for only a small subset of the population. And then when you get back into brain damage, then we almost have no treatments whatsoever. We have at the McPherson ERI Research Institute folks that are interested in every one of these aspects of our visual system, both in terms of trying how to understand it better and also developing therapeutics. So how do things go wrong? So clearly, we understand trauma. If somebody has, an explosion or a gunshot wound or something like that that disrupts pathways, we understand that. But there's actually a lot of ways things can go wrong in such a delicate system. And I'm going to start with looking at the eyeball. So what I'm showing you here now is a blown up version of a cross section of the eye, showing on the left all the cornea and the lens, all the focusing aspects of it. And then on the far right, you see that there's an orangish-like structure, and that's the retina, that thin film that attaches to the very, very back of the eye. So what fills up the rest of it? Something called the vitreous humor, basically a clear jelly, and that helps to maintain the shape of the eye and the optical clarity of the eye. If we look at the retina, though, if you just look at it, the retina itself doesn't look like much. It looks like a thin film, much like saran wrap. But if you look at it under a microscope, it's actually very, very elegant. It's got multiple different layers of what we call cells, like a circuit. So just like when you turn on the light, you might have a wire that goes to your electrical box, and that electrical box goes out to the curb, and then that goes on to the power stage, so on and so forth. The retina is arranged like that too. So the deepest layer within the retina are where the photoreceptors live. And those are your rods and your cones. They're responsible for detecting light. But they can't detect light entirely on their own. They need helper cells called retinal pigment epithelium. They're responsible for giving nutrients and other things that are necessary for the photoreceptors to work, and also taking away waste products. And if the photoreceptors and RPE work in tandem correctly, then it generates a signal. So it turns light into a chemical signal that is then passed along to a way station cell called a bipolar cell, which then passes it to a third cell called a ganglion cell. There are 1.2 million wires through the optic nerve back to the brain, which actually does the seeing, the perception, which is done in the brain. So I'm progressively blowing things up. It's almost like you're zooming in on these cells from the outside of your head into the eye, into the retina, and now looking at what a stylistic impression of a photoreceptor cell. So one of these cone cells, for instance, that's responsible for detecting red or green light, it functions because it has special proteins within it that are specially designed to detect light and convert that light signal into chemicals that other cells can understand. Because no other cell in your body can understand a photon of light. It has no idea what to do with it, but your photoreceptors do. But it has to translate that into something that the other cells in the retina can understand. And those are chemical or electrical signals. To do that, it has lots and lots of genes. So it has this blueprint in its nucleus. So we all have DNA, and that DNA codes for thousands and thousands of proteins that help each and every one of our cells function properly. And that DNA tells the cell exactly how to make these individual proteins. And those proteins, again, tens and tens of thousands of them, are what ultimately do the function of the cell. Now, unfortunately, in some folks, this intrinsically can go wrong. So if there's, for instance, a mutation in one of these blueprints, such that there's a glitch, so that the cell no longer knows how to make a protein, and if that protein is very important for a function, say like detecting light, the cell loses its ability to do that function or detect light. And the funny thing is that when cells lose their function in life, oftentimes that can lead to the whole cell eventually degenerating. And then we're in a real pickle, because not only are we missing the protein that's necessary, but we're missing the whole cell. So it's sort of like saying, well, my furnace went bad, but over time, without a good furnace, the whole house gets into disarray, and then the house sort of crumbles down. A lot easier to replace a furnace than it is a whole house. Now, the other way that this can go wrong is not have it be the cell's fault or the DNA's fault, but if there's an external injury. So if something happens through no fault of the cells itself, let's say inflammation and inflammatory disease, pressure from a tumor, or an injury from outside, say from a stick in the eye or something like that, that also can make the whole system go awry, of course. So when should we intervene? So it's not just a matter of what goes wrong, but we also have to pay attention to where along the process is a person who's experiencing vision loss. So early on, when everything is hunky-dory and there's no problems whatsoever, on the far left here, I have that as normal. The stylistic cartoon of the circuitry of the retina, in red being the photoreceptors and in blue at the top being the ganglion cells, everything's great. In that situation, we want to preserve things because it's easier to keep what you've got than to replace things once they've become dysfunctional or been lost. So at that point, we want to focus on prevention, for instance, exercise, diet, things like that, and perhaps pharmacologic treatments or drugs. Later on, during the course of a disease, you may start to have critical loss of proteins and function, but the cell is still there. In that case, you could be able to intervene by putting in healthy copies of genes to hopefully get the cell to regain its function. But further along in disease, the cell entirely degenerates. Then you have to look at ways to replace the cell or bypass it, perhaps with an electronic chip. And then in the far end of the spectrum, things can be really discombobulated, where you have multiple different cell types that are gone. intense scarring, or perhaps the entire organ is not working anymore, in which case you have to replace not just one cell or two, but whole tissues. So the bottom line comes down to is when should we intervene? As early as we possibly can. Unfortunately, a lot of these diseases don't have early interventions. And so what we're trying to do is meet everybody where they're at. So we want to work on ways to prevent the disease or the injury from happening, as well as trying to slow it down or maybe reverse the effects of the disease so that nobody gets left behind. How do we do that? So we can do that, as I mentioned, through prevention or drugs. We can do it through manipulation of genes, trying to correct or replace the genes that aren't working well in folks with genetic disorders, replacing the cells once they're lost, or bypassing them in some way, or in the far future, perhaps using prosthetics that actually work on the brain directly, or replacing whole tissues or organs. So that brings us to how things are going. And I get this question all the time. So this is all fine, well, and good, but where are we in this whole process? And I start off always by saying, well, things never go fast enough. If you're experiencing vision loss, 1 hour is too long to wait for a therapy to come across your Google alerts. But then there has been significant process in multiple areas, and folks that have had vision loss for a longer period of time will remember not just very long ago, 10, 15, 20 years ago, when nothing was happening. And there was very little research and very, very few, if any, clinical trials going on. And not every clinical trial, not every effort is going to result in a miracle cure or a cure at all, or even a decent therapy. But the fact that we're having more and more shots on goal means that we will continue to find ways to improve the condition for folks undergoing vision loss. So I'm going to focus on 4 areas: drug therapy, and give you an example, 1 or 2 examples in each one of these areas, which hopefully we'll touch on a few. So drug therapy would be 1, gene therapy would be the 2nd, cell therapy and therapeutic delivery. Okay, so we'll start with drug therapies. So the issue with drug therapies is that they're convenient in that you may be able to take them by mouth, which is nice, but they've got to travel through your entire body and cause effects just on your eye and not on everything else, and not have side effects elsewhere. So it turns out it's really difficult to be able to take an oral drug, or even by vein, and get it to get where it needs to be so it can get into the retina. The eyeball actually has ways to kind of block drugs from getting into it. But people are smart and they find ways to kind of Trojan horse those drugs into the retina. So I'm going to give you one example, and this is for a disease called Stargardt disease. And so what I'm going to show you is, I mentioned before that the photoreceptors work very intricately with their helper cells called the retinal pigment epithelium. And one of the real key things that it does is there's a special chemical in the photoreceptor that has to change shape when it gets hit by a photon of light. When it does that, it changes shape and initiates a signal that ends up in us seeing something. But then it has to get recycled. That chemical has to get recharged. Think of it as a recharged battery. And that occurs by getting cycled back into the helper cell, where it gets recharged and then placed back into the photoreceptor. Unfortunately, in some diseases like Stargardt disease, which causes central blindness, starting in young people, that system gets gummed up. And what happens is that one particular step in this very complex biochemical pathway no longer works. And that leads to a buildup of waste products called lipofuscin. And those waste products build up and act kind of like a poison pill. And so what folks are doing, and there's a particular company called Alkeus Pharmaceuticals, as well as others, are working on ways to make new chemicals that are not susceptible to getting gummed up, so that the system can work even though the proper machinery isn't quite there. And these are now in clinical trial, and they're showing some benefits. I think you'll find that one of the themes of what I'm going to say is that there's no miracle cures. There's no way of snapping your fingers and making it all go away. But some of these therapeutics can help damp the effect and prolong periods of better vision for patients. A lot of them can't reverse the effects of the disease, though. But this is brand new work, and it's in phase 2 trials, so it's showing some promise. It may also have some benefit in folks with macular degeneration as well. So Stargardt disease, that's great. If you have Stargardt disease, you're going, oh good, there's something out there for me. But Stargardt is just one of hundreds of different inherited diseases. And so it's difficult to kind of contemplate having to make specialized treatment for each and every one of them. Although that is happening, people are taking individual genetic disorders and developing therapies for each one. But it can be frustrating for people when they don't see their particular disease on that list. So it's also important to be able to develop more broad-based disorders that can affect more than just one type of disease. And so one way to do that is to do it through gene therapy. Now again, gene therapy is designed to go after one, perhaps more than one type of genetic disorder. But to kind of get you a sense of what goes on during gene therapy, I'm going to give a little bit of, hopefully not too detailed, genetic background. I mentioned before that we have DNA, and those DNA are the blueprint for every protein in our body, which has to be working normal for things to function well. We have two normal copies of every gene. So we have a backup copy for every protein that we need to make in every cell of our body. And when both of those are working fine, we have great function. Okay? In some cases, called recessive gene mutations, there are problems in both of the genes. So both pairs of the genes have something in them that cause the protein not to be made. So basically, you have an absence of a critical protein. As you might imagine, if that occurs, the protein doesn't function, and you have some sort of a disease that manifests itself. One example of this would be Leber congenital amaurosis. Dominant gene mutations actually only affect one of the two genes, but instead of not causing the protein to be made, it actually creates a bad actor. So one of the two genes, the one that has the mutation, makes a bad protein that messes up the function of the good protein. And an example of a dominant disease is something called Best macular dystrophy. The reason I bring this up is to illustrate the point that not every gene therapy works for everyone. So it depends on what type of gene mutation you have, where you are in the course of the disease. Do you have recessive or do you have dominant? So how are we trying to address this extreme variability in different ways that people go blind by genetic mutations? Well, Dr. Bakash Patnaik of the Department of Pediatrics here at UW is looking at the recessive diseases like Leber congenital amaurosis. And what he's done is he's developed a way that he can deliver good copies of a gene, in this case, a disease called LCA16 that affects little children. And in this case, most of the people that have this disease all have the exact same mutation. So he can develop one single therapy, and he worked with a team of investigators, including our laboratory, that made stem cell lines from children who had this disease so that we could study them in a dish. He also made a mouse model that had this same disease so that he could test a way in which he could deliver healthy copies of the gene to these cell types in a process called gene augmentation. So essentially, you're delivering to a cell that no longer makes an important gene healthy copies of that gene. He does that by a direct injection. So you have to actually directly inject that gene using a virus, and the viruses have a very bad rap for many good reasons, but the viruses that are used in patients are crippled such that they can't cause disease, but can deliver their payload to the right place within the eye. And so by doing this, he's able to deliver healthy copies to the retina. And what he showed was recovery of all the patient's cells and the animal models for this broad-based type of disease. So it's very exciting. And very recently, in the last few months, a company has taken over a license for this technology. And hopefully in the next year or so, we will have the initiation of clinical trials for these children that are going blind from Leber congenital amaurosis from this gene defect. So very exciting development for gene therapy. Similarly, a different disease called Best macular dystrophy, in this case, it's a dominant disease. So normally, you can't just say, well, we're going to deliver healthy copies, because you have to deal with the bad copy of the gene at the same time. But a big McPherson ERI team, and this was actually, if you look at the news feed for UW-Madison today, this was just put out as a news lead from the University of Wisconsin-Madison. And it was a large team with Bakash, myself, and Dr. Saha in engineering, and Dr. Roy in biostatistics and medical informatics. And Best disease is a disease that causes central blindness over time. I'm showing a picture of what the retina looks like. It's got this big yellow lesion where a lot of waste products build up. And the folks that actually do the work in the lab, I got to give them credit. Dr. Sinha and Dr. Steyer and Shahi were also very instrumental in this study. And what we did with Best disease is we took patients who had the same disease, but multiple different genetic mutations. And what we showed was that using stem cells derived from each one of them, and here's one family that has Best disease. The gentleman on the left was a former truck driver who lost his job because of his loss of vision. And we made his own retinal cells in a dish and tested a special type of gene therapy, much like what Dr. Patnaik was testing, and found that by just simply swapping out the bad copy of the gene, that we could restore the function of the gene in almost all of the patients, but not all of them. So what do we do about the folks that don't respond? Well, we turn to Dr. Saha here on campus, who does a special type of treatment called gene editing. So instead of just putting new good copies of the gene into the patient's eyes, we look to actually specifically fix the genetic mutation in the cell types. And this was successful in correcting all of the patient's defects or in their cells that did not respond to the first line treatment. And so in this way, we hope to have something for all folks with Best disease to turn to when it comes to therapeutics. And like Dr. Patnaik's therapy that I showed you earlier, a company now has taken on this challenge, and we hope in the next 1 to 2 years to have gene therapy for Best disease. Both of these conditions, by the way, currently have no treatments. And so we're very proud of the McPherson ERI to play a prominent role in the advancement of therapeutics where there were none, even in the recent past. Okay, so that's fine and good for people who still have some function left and some cells left to rescue. But what about folks where the cells are all gone? And in that case, what we concentrate on here is replacing those cells through stem cell therapy. And now that's something near and dear to my own heart. So in cell therapy, the question becomes, where do we come up with a source of cells that can replace the cells that have been lost in the course of disease? And the nice thing about cell therapy is it's not an easy thing to accomplish, but if you can accomplish it, it's relatively agnostic, or it doesn't really care how you lost your cells. So in other words, you could have lost it from retinitis pigmentosa or macular degeneration or some other process, but if it ends up with the cells dying, doesn't really matter. We just want to replace those cells with healthy cells. But to do that, we have to manufacture human retina. And so we were the first to do that here with human stem cells at the McPherson Eye Research Institute, being able to grow in a dish from patients' own skin cells or blood cells, retinal tissues. And those retinal tissues go on, I'm going to show you here, to make all the cell types that I showed you earlier, the photoreceptors, the rods and the cones. And they can detect light, and they can transmit that light signal into a chemical, which hopefully then can stimulate other cell types, including cell types of patients that are still functioning, so that we can get them to see again. And this is I'm showing you an example of a highly magnified view of what some of those cells look like. The red dots that see there, if you can see them on your computer screen, are thousands of cone cells, and the green dots are thousands of rod cells. And as you probably have all heard, or many of you have heard, this has spurred the foundation of a company here in Madison called Opsis Therapeutics that works hand in hand with Fujifilm Cellular Dynamics International. And the hope is in the next year, 3 years, to have clinical trials for folks that have lost photoreceptors or lost both the photoreceptors and the retinal pigment epithelium in diseases like retinitis pigmentosa and macular degeneration, so that we have a treatment for them in the foreseeable future. And then, of course, you can combine therapies. And folks that are doing that are like Dr. Akita, or Aki Akita, who's in the Department of Medical Genetics and is the Timothy William Trout Chair in Eye Research. And what Dr. Akita does is he's interested in a particular part of the cell called the mitochondria. And the mitochondria are the powerhouses of the cell, and they're involved in aging and also in age-related macular degeneration. And so he's done some magnificent work looking at specific ways in which the cell maintains energy states, which may go awry in disease. And this is a really incredible but quick video, which shows one of the proteins that Dr. Akita discovered, how it goes and binds to and affects the structure of mitochondria. So what I'll show for those who can see it is a purple dot, and that purple dot zooms in attaches to the mitochondria and splits it. And this splitting of mitochondria is important in its normal function. And so when these proteins don't work well, it affects how the mitochondria work and how the retina ages. And so he's working on ways both via gene therapy and drugs to try to intervene and make retinas healthier for a lifetime. All these therapies are well and good, but if we can't get them to the right place safely within the eye or the visual system and do it effectively, then it doesn't do anybody much good. So we have engineers and scientists whose job it is not necessarily to make the therapeutics, but to develop ways to get them more effectively where they need to be to do their job. So Dr. Gong is just one of many of our engineers in the McPherson ERI Research Institute, and she's a specialist in nanoparticles. So she's able to take these therapeutics, which the body would otherwise degrade and get rid of and reject, and coat them in these nanoparticles that allow them to evade the body's attempt to get rid of them, so that they can pass into and do their work inside the cells where they're needed in the eye and visual system. This is very exciting work that was published in one of the best journals in medicine. And in addition, I'm very thankful to work with Dr. Gong and Dr. Ma, who's a computer engineer and electrical engineer, along with people at my lab, Dr. Phillips and Ali Ludwig, to develop patches. So I showed you earlier that we can make these rods and these cones. That's great, but how do we get them where they need to be? Well, Dr. Gong and Dr. Ma can fashion these tiny, tiny little scaffolds or patches made of biodegradable material that we can subsequently put our photoreceptors into, which allows them to line up and allows our surgeons to precisely place them into the subretinal space. So this is a video of one of our collaborators at the National Eye Institute putting one of these patches into the subretinal space near the visual streak, the equivalent of a macula, in a pig. And so this is part of all of the efforts that we have to bring this to the clinic in a timely fashion. Okay, well, I hope I didn't totally lose everybody and inundate people with too much information. It's a delicate balance between giving a little bit of an update and giving too much. And so your feedback will be important after we're done today so that we can do this better in the future. But at this point, I'm going to kick it back to Michael, who hopefully has some questions for us from you folks. I also have questions that were submitted ahead of time that I can answer as well. But you guys have heard my voice plenty of time, so I will kick it back over to Michael.
Thanks, Dave. So as everyone knows, there are many, many researchers both here and elsewhere working on different therapeutics, and to cover everything would be impossible in one meeting or even many. So we really want your questions to drive what we talk about for the next hour or so, or as long as we have questions. We do have half a dozen people who ask questions in advance, and while people are sending in their questions, we can start with a few of those.
Okay, we had a couple of questions related to glaucoma, and I know I touched on that a little bit, but also a lot of what I chose to talk to you about was relatively heavy on photoreceptors and retinitis pigmentosa. So I thought it was good that we had a couple of questions with regard to glaucoma. I would be remiss if I didn't mention that one of our advisory board members is Dr. Harry Roth, who, as many of you know, he provided glaucoma care for many, many years here to the people in Madison. So we're very happy to have him on. So if I mess up, I'm in trouble. But, but Harry, please feel free to chime in as well. So new information regarding research on glaucoma. So in general, and this is a great question, glaucoma is a complex disease, and it actually isn't one disease. It's many, many different types of disease. And that makes it hard when people say, is there a therapy for glaucoma? Because again, it kind of depends on what type of glaucoma you have. And as I mentioned before, the vast majority of people with glaucoma do have effective treatments, but a percentage of them fall through the cracks with the therapies that we currently have. So what can we do for those folks that aren't maintained well with current therapies? One way that people kind of fall through the cracks is that the drops that are used or the surgeries that are offered aren't enough, and they continue to have high pressures in the eye. And those high pressures cause damage to those ganglion cells or the optic nerve. And I have a slide here to show you. So in glaucoma, you have, I'm showing on the left here, a normal optic nerve. It's kind of hard to see, But the optic nerve should be chock full of 1.2 million very healthy wires. And if it does that, if it is, then it looks kind of orange and healthy. But as those ganglion cells, those wires die back, you start to see what looks like just kind of a pale ghost of what should be there. And that's shown on the right. And the ganglion cells are those cells that are kind of not directly connected to the photoreceptors, but are a couple of cells away from it in the circuit. And unfortunately, if you have very high pressures, or sometimes if you have normal pressures, but what you might think of as weak ganglion cells, those will degenerate over time. And in that case, it doesn't matter if your photoreceptors are working just fine. You've disrupted the circuit downstream of it, so there's no image getting back to the brain. And so the point there is, can we keep the ganglion cells happy? And we do that by trying to lower pressure if that's the problem. And there are always new ways to do that. New drugs are being developed by folks here as well as elsewhere around the world to try and help those who aren't responding to normal drops to lower pressure. There's also gene therapies now being developed by people on campus like Paul Kaufman, and we have other great glaucoma researchers, including Rob Nichols and Dr. McClellan, and the list goes on. We actually had an Insights newsletter that focused on glaucoma recently. And these gene therapies are trying new ways, so not just different types of drugs, but actually can we use gene therapy to help with the eye's ability to drain fluid? And if we can do that, then perhaps we can help those who aren't responding to drops. But then there are folks that no matter how low you get their eye pressures, they still go blind. And these are called normal tension glaucoma patients. And these folks, we can't just lower pressure or take the pressure off the nerve or the ganglion cells. We actually have to try and make the ganglion cells stronger so that they can resist normal or even high pressures. We do that, people are trying to do that with what are called neuroprotective agents, or drugs that actually make the ganglion cells less likely to die. And for instance, Dr. Robert Nichols here in the McPherson ERI Research Institute and at UW works on drugs that hopefully can help that happen. And then lastly, if all of that fails, what about replacing ganglion cells? Can we actually go back and get those cells to hook back up again? Well, there is a large effort, a national effort of which we're participating called the Audacious Goals Initiative from the National Eye Institute that's trying to do just that, regenerate ganglion cells. But it's a very, very difficult thing to do because as opposed to replacing photoreceptors, which just have to make very local connections, ganglion cells have to reach way back into the brain. So as you might imagine, that's a much taller order and a more difficult thing to accomplish than other types of cell replacement. That being said, what was a pipe dream 10, 20 years ago is something that millions and millions of dollars is being poured into now because people think it can work. Are we close to it now? No, unfortunately we're not, but it is on the horizon and a lot of very, very smart people think we can accomplish. So getting back to the second question there, it's sort of going along the lines of what I already talked about, but we had another person online today who's been unfortunately dealing with glaucoma for over 30 years and has not been responsive to drops. In fact, has developed allergies to drops, nor has surgery really been able to help that person. And so that person falls into that unfortunate bucket where, you know, we don't really have anything good for him or her right now. And so again, the question is, are there any miracles on the horizon? And I bring it up to say, probably no miracles. I'm a firm believer in miracles myself, but I don't think we control them as scientists. But in terms of what we can develop, I think there are therapies that are being produced each and every year. They don't work for everybody, and they're not home runs out of the park. But I think that the more and more therapeutics that are being developed, the more likely that we're going to hit on one that's going to be good for everybody. So there's a lot of hope out there, but there's also, you know, with a dose of realism as well.
We've got a couple of questions from Lynn Van Vriede, Dave, and I'm going to ask them both because I'm not sure whether they're related or not. But first question is, how important is it to maintain low inflammation? And is eye inflammation the same as general inflammation in your body?
Again, great question. So in many respects, you know, we think of things like if I have an inflammatory disease, is that affecting all of my organs equally? And I think people that are dealing with that know that that's not the case. If you have arthritis, your knees are bad, but your liver is okay. So inflammation has a way of selectively choosing what it wants to deal with. And some of them are more indiscriminate than others. And it can be very insidious as well. So the eye, in fact, is a target for many different types of inflammatory disorders, but not always. So for instance, rheumatoid arthritis affects the knees and sometimes can affect the front part of the eye, for which there are oftentimes very good therapies, oftentimes the same therapies you take for your knee because they're systemic and they go throughout your whole body. So the answer is, in broad terms, yes. So in general, we try to avoid inciting inflammation in our bodies, and that's where a good diet comes into play. If you happen to be on immunomodulatory agents or anti-inflammatories for other diseases, there may be secondary good benefits on your eye. But we have to be careful. We don't want to take drugs that have potential side effects too. Some medications that are designed to improve inflammation in your body could have negative effects on your eye too. So don't take medications, obviously, kind of on your own, just thinking, well, if it's good for the goose, it's good for the gander. Make sure that you're talking this over with your eye care specialist. But a good diet, exercise are all very important to just stay in general healthy, and that's good for your eye as it is for the rest of your body.
And Dave, there's a follow-up from Lynn. Is there a way to repair a retina damaged from Plaquenil used for RA?
So Plaquenil is a type of an immunomodulatory agent. So that's something you take for any number of inflammatory diseases. It's also called hydroxychloroquine, which you may have seen in the news because it was all the rage to cure COVID for a while. So Plaquenil has been around forever. It was initially used to treat malaria, but was found to have beneficial effects in suppressing the immune system. So it's used for lots of different things, including some types of arthritis. But it can cause direct damage to the macula. So it can affect the photoreceptors and retinal pigment epithelium in the center of the retina. It's rare to see that. It was seen many, many decades ago when people didn't understand the proper doses to use, and when there was a predecessor of hydroxychloroquine called chloroquine that was used. And so unfortunately, that caused degeneration. That's an iatrogenic or a drug-based cause of death of photoreceptors and central blindness. No one wanted that to happen. They wanted to control the inflammation, but there was this unfortunate side effect of blindness because of its use. So Plaquenil is used all the time. I have tons of patients that are on it, as do all ophthalmologists, and we watch them closely to make sure that they maintain good vision. And if they show the slightest sign that they're losing vision, we get them off of it. Once the damage has occurred, though, then that falls into the category of needing cells replaced. And so that would be on the radar for cell replacement therapies like I talked about. Probably not first in line, but it's a possibility that some folks may be able to be helped by that. So again, great question, and thanks for putting those two together. I swear I didn't set her up on that one.
Okay, we have a question from Jim and Ellen Hunter. As an older person with very little vision, I'd like to know what the prospect is for clinical stem cell trials for syndromic RP, Bardet-Biedl syndrome.
Okay, Bardet-Biedl. Okay, so first of all, from a vision standpoint, it doesn't matter if it's syndromic or not. So when we're talking about therapies, whether it's cell therapies or gene therapies, they will be delivered inside the eye. And the intent is for that therapy to stay in the eye. In fact, the FDA does not like it when your therapy leaks out into other areas. So even though you may have other effects of a genetic disorder beyond just blindness, the types of therapies I'm talking about, for the most part, will only affect the blindness portion of it. And I know you know that, but I thought it'd be good for the rest of the audience. So cell therapy for something like Bardet-Biedl would essentially be the same as it would be for other types of retinitis pigmentosa, which is the good part about it. The good part is that while it's not going to be a cure-all, certainly it's not going to be a cure-all, but it's not going to be a therapy for everybody. It's certainly going to touch many more people at once than a gene therapy would. In your case, whether it be syndromic RP, Bardet-Biedl, or other types of RP, you've got good company. You're all in that same bucket when it comes to cell therapy. Now, the downside of cell therapy is you have to be later in the disease to benefit from it. So ideally, you never get to it. I tell folks, even though I do cell therapy and I do stem cell therapy, my hope is nobody ever needs what we're trying to make, because that means you get to keep your cells that are working functionally and well, and you don't have to look to replace them later on. But if that does happen and you do, plinko your way into a situation where the cells are lost, then we hope to have something that's available for a lot of people and not just one or two subsets of the population.
Okay, Dave, do you want to take one of the questions that you have in advance?
I'll switch back over to my screen here. Okay, another question that we got was, and I like this one because it's a clinical trial-based question. We could spend, and maybe we will, if you guys are willing to do this again, maybe we'll focus on clinical trials. What are they? What types of clinical trials are there? How do you get enrolled in one? What are the pluses and minuses of it? Why do they move so darn slow? When's mine coming? That sort of thing. But I like this question because it's clinical trial oriented. And you'll hear this. So there's a clinical trial that in Europe or Asia, and it's looking to do something called remove lipofuscin. Remember, I've mentioned waste products a lot in the retina. That yellowish material that you see in dry macular degeneration, or that big glob of yellow stuff you saw in Best disease that I showed. So there's some thought that these waste products themselves might be detrimental, and that's debatable. A lot of scientists believe that it's a chicken or the egg thing. There's something that goes wrong, it leads to waste products. The waste products don't cause the thing to go wrong. But that being said, nobody's leaving any stone unturned. We want to try and go from every possible angle. So people have looked for ways to try and get rid of these waste products to hopefully slow down the process of disease, whether that be AMD or Stargardt or other types of diseases. What this participant is saying is that apparently little or nothing is being done in the US. So firstly, there's a frustration there, obviously. Why in Europe and Asia and not in the US? Is the US just being stubborn? And this gets back to how clinical trials are put together. The answer is no. It actually is the decision of the company doing the trial. So if it's a European company, and that European company has licensed the product, they have exclusive use of it. They can determine where they want to have the clinical trial. Same thing happens in the US. There are people in Europe and Asia going, why are there trials in the US and they aren't in Asia or Europe? So those sorts of things are dependent upon the people who are setting up the trial. And they can't initially put it everywhere because the various regulatory agents need to show that it's safe. So they can't just take a brand new product and just throw it to the wind, because what if it causes damage? So they have to keep it small at first, what's called a phase 1 trial, in very localized areas around the world. Once it demonstrates safety and effectiveness, then they can expand it to other areas. Believe me, every one of these companies wants to make money. Every one of them wants their product to be available everywhere. So there is no conspiracy about just holding it in one spot. Now, there are some countries that are a little looser about clinical trials than others. And so, but that's not necessarily a good thing either. And the US really, and the FDA, has shown itself to be very, I believe, reasonable and intelligent when it comes to advancing clinical trials. And we are seeing clinical trials go much faster now than they did in the past. The first gene therapy in the retina, for the retina, the Luxturna drug, took over a decade to progress from there being no gene therapies to being therapies for everybody that had that particular rare genetic defect. Now we're seeing turnarounds in a year or two. So again, a year or two is more than somebody who's going blind wants to hear, but it's better than a decade. So to finish off this question, would removal of lipofuscin enhance the success of implanting stem cells in the eye? It may, and it brings up a good point. When you're putting new cells into a diseased retina, you still have the effects of the disease that are there, or perhaps even the ongoing disease process. So it's like putting new wine into old wineskins. It doesn't really work if the wineskins burst. So the idea of next generation therapeutics trying to better prepare the patient's remaining retina so that it can make those connections better, that's an important next step and things that we're working on already now, but that we hope will be an improvement on the existing technology. That's the stepwise process that occurs in the development of anything that's new, whether it be airplanes or computers or what have you. You can remember not too far back where computers filled an entire room and could barely add. And now we've got supercomputers on our wrists. But it didn't happen overnight. It took trial and error and people innovating over time. And that's what we're here for at the McPherson ERI Research Institute, to look where there are gaps, where there are no therapeutics or not good therapeutics, make them better, invent new ones, and then not just rest on our laurels, but continuously innovate and make them better.
Dave, we actually have two questions on macular degeneration that I believe are related, so I'm going to give you both of them now. First, from Margaret Potratz, is there a general timeline on when a person is diagnosed with wet AMD to when their vision becomes very low? And the second question from Joni Shahrani, my mother has macular degeneration, and recently she has begun to see wavy lines when looking at pillars. Can you explain what is happening with her vision, and does she need different treatment? Currently, she is receiving shots in her eyes.
Okay, well, I'm actually going to be able to get a couple birds with one stone here. So I'm going to move along to macular degeneration. So that's another one of our questions that we had that was submitted. So I'm going to put this slide up here just to basically kind of let people know what do we mean when we talk about dry AMD or age-related macular degeneration, or wet AMD or age-related macular degeneration. And the difference is pretty simple. In wet AMD, there's bleeding. The macular degeneration has caused abnormal blood vessels to grow under the retina where they are fragile, and then they break and they bleed. And so what happens when the bleeding occurs, as you might imagine, is you have an acute vision loss. So in dry AMD, it's kind of a slow smoldering process. We don't understand it entirely, and we have a large group of investigators here in the McPherson ERI Research Institute who are looking into this disease specifically because it's so prevalent and because there's so much that still needs to be done to understand it and to treat it more effectively. But in dry AMD, you can think of it like a slow burning forest fire where the retinal pigment epithelium and the overlying photoreceptors slowly die off from the center outwards. Now, it doesn't progress to full blindness, it only affects the center. So I tell folks with macular degeneration that they're not going to go completely blind if all they have is macular degeneration, but it does rob you of your most precious vision, which is the central vision that you use to read, to drive, to recognize your grandchildren's faces, and so on and so forth. In dry AMD, you have loss of those cells, so it's one of the prime targets for cell therapies because we want to be able to replace them. And below the pictures of the retinas, where the geographic atrophy or the area of cell loss is shown as this kind of lighter spot, you can see that there's a loss of, in this underneath, it's called an OCT. It's a cross-section of the retina. So there's a critical loss of stuff under the retina. Now, if there's bleeding, though, you'll see that it tents up with all the blood that's underneath it. So on the right side, the retina has gone from sinking down because it doesn't have cells to poofing up because now it's filled with blood like a blood blister. So you can be slowly losing vision from dry AMD, and then all of a sudden you experience an acute loss of vision because this blood has occurred and it's now obscuring the light from being able to get into the retina and let your central retina work well. If that happens, what it tells you is that there are these abnormal blood vessels. And fortunately, and this wasn't the case again 10, 15 years ago, but there are injections that can help dry up the blood part of it. Not in everybody, but in a lot of folks. So people who have wet AMD, which you don't want to have because you've acutely lost vision, but people who have had that more advanced type of macular degeneration, sometimes and maybe oftentimes can respond to these injections that help to dry that up. But when you dry it up, you're still left with the macular degeneration that was there before the bleeding occurred. So it doesn't cure macular degeneration. It doesn't get rid of the slow-moving train. It just gets rid of the blood that caused the acute vision loss. So believe me, people who have wet AMD are thrilled because they can regain vision once that blood clears. But it doesn't affect the slower course of that dry, smoldering forest fire that occurs.
Well, what I'm hearing from that is that there's probably not a general timeline when a person is diagnosed to when their vision becomes very.
So yes, so that's a common theme with all macular degeneration. It's quite person dependent. There are plenty of people who are told that they have a little bit of dry macular degeneration, that have some little yellow spots in their retina, and they live their whole life and never really lose appreciable vision. And the older we get, the more likely we are to have that. In some respects, macular degeneration is kind of a normal aging process. If we all lived to be 150, we'd probably all have macular degeneration. It's just kind of how our parts wear out. The question is, why do some people's parts wear out when they're 50 and some when they're 110 and some never? And so we're trying to understand that. So yes, it's hard to predict. There are large groups of people that are following individuals with macular degeneration, including folks here at the University of Wisconsin-Madison, like Barb Blody and other retina specialists who look at hundreds and thousands of photographs of individuals with macular degeneration and trying to understand, are there tells, are there certain things we can look at that will tell us whether or not that patient's going to go on to lose vision, or do they have a lot of years left in them? And that's important because if we know that, we can then categorize those people for different types of therapy. Obviously, we don't want to touch the person who's going to be fine their whole life or has 20 more years of good vision. But we want to be more aggressive with people who show signs that they may be going downhill faster. And of course, bleeds can happen anytime. So those are surprises to everybody when they happen. But like I said, thankfully, there are some people who benefit from shots. But there are some people who don't. And those people that don't are like the glaucoma patients I mentioned earlier. And we're hoping to develop therapies so that they have something for them. And there are stem cell therapies now for wet AMD, but only 2 patients have been treated thus far. And one of them showed improvement. And so hope that trial will be started up again in the near future.
Dave, on the question of what is specifically happening when the patient
Wavy lines.
Wavy lines.
Okay, so this is a great question. And so what you are given, all people with macular degeneration are given what's called an Amsler grid, which essentially is super fancy and is basically a piece of paper with vertical and horizontal lines drawn on it. And they ask you to look at the center and to report to your ophthalmologist if any of them look wavy. So why does this give us a sense of what's going on in the retina? And that's because, if you remember, the retina is like a film. It's a thin film in the back of the eye. And it's important for it to work properly to be flat up against the back wall of the eye. If it starts to pooch forward or it starts to get a little bit wrinkly, then you see, as the person looking out from those eyes, the world kind of get wrinkly or get kind of wavy. So when people look at a pillar or people look at the edge of a wall, they may see it, parts of it missing, or they might see it wave in and out. And so that's a subjective way or a way of the patient monitoring him or herself to say, yeah, something's going on in my retina. Now, are there other things that could be causing that? Yes. That's not the only thing that causes waviness of vision. Things called epiretinal membranes also do too. But anything that causes distortion of the retina in the center of the retina can cause those wavy lines. So it's an important way to monitor your retinal health. And of course, if you do see changes, letting your retina specialist or ophthalmologist know.
And is it correct to say there's no other treatment, no alternate treatment at this point other than the shots?
Yes, that is correct. So the dry portion of that, the smoldering forest fire, is one of the prime targets here at the Eye Institute and for stem cell therapy. There's also people working on drug and perhaps gene therapy. It's interesting because macular degeneration is not a one gene disorder. So it's not like you can inherit a gene and you know you're going to get macular degeneration. It's more like diabetes in terms of it runs in families. So if your family has multiple family members that have it, is it more likely that you might develop it? Yes. Does it mean you will develop it? No. And so we're getting a better handle on what are the various environmental as well as kind of genetic components that predispose people to getting it. But even that right now, is not extremely well understood and is a topic of study here at the Institute and around the world.
So we have a couple more questions that have come in about AMD. So let's on that for a moment. From Kristen Fontaine, is the dark adaptation test a good early predictor of AMD, and should more aggressive interventions happen sooner to help prevent further degeneration to the macula?
Good question. So that's actually an entirely different topic that I could have put up here. I'm glad you brought it up. So I mentioned the therapies and I mentioned the delivery of the therapies. Another huge aspect of the Eye Institute and the research that we do is better understanding what exactly goes wrong in the first place. Because if you don't understand the mechanism of the disease, then we're kind of just shooting in the dark with regard to therapies. So the better we understand what goes on, the more we can be smart about zeroing in on how we want to intervene. So one of that ways is diagnosis. So if we understand the disease better, we can diagnose it better, more accurately, and hopefully earlier. So dark adaptation, basically what this is not that simple of a test. So when people develop tests, the ideal test is very accurate, almost never wrong, so you don't get false positives or false negatives. So every test that you have, there's a % chance that it will say you've got a problem when you really don't, or it'll say you have no problems and you really do. So obviously you want to minimize that. And so that's one of the aspects of trying to make the tests better. And then you want them more and more sensitive so that you can reach back earlier and earlier in the course of the disease so that you can identify that a person might have it and hopefully intervene sooner. So for macular degeneration, the dark adaptation test, what you're doing there is you're putting people in the dark or you're taking people from bright light and putting them in the dark and then looking to see how long it takes them to be able to see things well. We've all had that experience on a bright sunny day in the summer where we walk into a dark room and we're totally blinded. And it takes us a while to warm our eyes to warm up to dimmer light, but eventually we're able to see. That time period, how long that takes, there's a normal and then there's abnormal. Sometimes it takes too long. And when it takes too long, it's indicative that there may be a problem with some of the cells in your eye. Now, I could get really complex, and I'm not going to. But there's data coming out to suggest that there's a certain part of the retina that's not in the exact center that may be going wrong first. So before you lose central vision, there may be a ring around the center that starts to be abnormal, and that can be picked up by dark adaptation tests. They're not easily done these days. Only specialized areas can do it. And the problem is, if they identify it, what do you do? So you could say, well, maybe you could start the vitamins. So the AREDS vitamins, of which, by the way, I will put in a plug for the University of Wisconsin-Madison. We were one of the prime centers that tested the AREDS vitamins and showed them to be at least, in some cases, very effective, some cases mildly effective in slowing down the process of macular degeneration. So you might put somebody on the vitamins earlier if you were to see that, but there's been no hard evidence to suggest that those vitamins help people at earlier stages. So the other aspect about these tests is if there's no intervention, if it's not going to change management, then insurance companies don't pay for it because they're in the business of doing tests just to do tests. So the diagnosis and the testing goes hand in hand with developing therapeutics. Because if you have a test that you can diagnose somebody earlier and then you have an intervention that will help alter the course of that disease, then you've got everything that is needed to be able to move it into patient usage. So in the meantime, all these things are being done on an experimental level, and hopefully these will meet up and ultimately be important for patient care in the near future. But again, it gives you a sense of how complex these things can be.
Dave, we have 2 questions about advanced AMD. From Lynn Van Vrede, how far along in macular degeneration can you still get help? And from Bob and Rita and Chrissy, with people who have advanced AMD, are eye vitamins such as Preservision and Ocuvite still effective?
So with regard to the first one, when is too late? Well, unfortunately, it's easy to get too late right now because there's not a lot that you can do. So there's the vitamins that one starts out when you have evidence of the onset of macular degeneration. Again, your ophthalmologist will tell you whether or not you are likely to benefit from them. I say that because the vitamins aren't cheap. People who are taking AREDS vitamins will know that they're not cheap. And so, you don't necessarily want to be taking them and dipping into your income if it's not going to benefit you potentially. So I'll just throw that out there. So, and then there are situations where once the cells are gone, I mean, the idea of the vitamins is to preserve the cells. If the cells are gone, then you're not going to benefit from those vitamins anymore. And then on the far end of the spectrum, where you have the bleeds, then you have the injections to dry up those bleeds. But in between, there's really nothing right now. And that's the huge gap that we're working on from the basis of developing new drugs, as well as cell therapies. So yes, the answer is that unfortunately, a large number of folks that have the dry type of macular degeneration or who fail to respond to the injections really don't have effective means of altering the course of their disease or restoring vision right now. And then the second one, Michael?
The second one was, are vitamins such as Preservision and Ocuvite still effective?
Yeah, so I think we kind of got at that with the previous question.
One more question from Carol Tollefsrud, and I apologize if I'm mispronouncing that, on macular degeneration. Is laser therapy still used for it?
Not really. So there's something called photodynamic therapy that was, I remember it being tested when I was a resident here back in the early 2000s. So there was a thought that low dose laser therapy might be effective at kind of drying up some of the problems that are some of the garbage perhaps that's accumulated in the back of the eye. And that really fell out of favor and did not show improvement. And so it's one of the situations where you have to take everything with a grain of salt. I mean, that wonderful therapy that you've heard about in Asia or whatever might end up being a bust. And as I'm sure everyone realizes, not every clinical trial ends up with something that works. In fact, a minority of clinical trials end up with therapeutics that are effective or are moved into market. But if those clinical trials don't happen, you have no chance. So we think of it like shots on goal. And as long as we're careful to be safe and thoughtful about the clinical trials that we're doing, then it's another opportunity for us to find something that does work. So if we're doing one clinical trial every 10 years, the odds that we're going to hit on something for everybody anytime soon is very low. But we're in the era now where hundreds of clinical trials are being done every year. And so that's more, darts at the dartboard. And so we will definitely see more and more therapeutics that are showing improvements. But like I said, it won't be fast enough. It won't be for enough people, but it will continue to move. And as long as we're careful and we're safe and we don't do any harm, we'll see an exponential growth in those clinical trials. We're seeing it already. The folks that have the deep pockets to pay for these clinical trials, which can cost $30, $40, $50 million to run one clinical trial. The people who can do that, the companies that can do that, are investing in the eye. They're investing in the retina. Why? Because the retina has led the way and has shown itself to be a safe organ to work in. So there are companies who really didn't start out even caring about the eye. Maybe they want help with the liver, or they want to help with arthritis in joints. But they can't start there because they don't know enough about those organs or how to get the drugs there. But because we know so much about the eye, they start there. So it's the gateway for a lot of drugs that are being developed. And that's wonderful for folks on this call and people that have eye-related disorders, because you get to be the first ones to see whether or not those medications, those gene cell therapies might be beneficial. We had a question about COVID-19. How has that affected us? And this is one of many reasons why I feel very fortunate to be here at the University of Wisconsin-Madison, where not only are we playing a big role in the fight against COVID, but I've got some very, very smart bosses. Our dean, our chancellor, the folks that run the university, I feel do a really good job of towing that line of being safe, but at the same time recognizing that the research that we do is very important. At its worst, back in March, April, there were things that were shut down, but a lot of labs were deemed essential. And so we were able to maintain some of our work that was critical, that if we were to have lost it, we would have lost years of research. We were not required to lose years of research. We had to jump through a lot of hoops and show that we were going to be safe about it. But those essential lines of research were maintained. And we're now up to, at least in my lab and many other labs, 100%. But we're doing so with social distancing, with masks, and with very strict rules. And we're learning to live with it, just like we're learning to live with Zoom and the teleconferences. So I feel very fortunate. I feel I've done a good job. Does that mean that there aren't going to be glitches along the way. No, a place as big as this is always going to have some glitches, but I feel very confident in our leaders to be able to navigate us through that. Now, getting to the question about federal funding, the National Institutes of Health, which is our primary funder for the research that's being done here in almost every university, has actually been increased. And I think that people recognize that it's kind of important to make sure that we keep the research engine running. Now, that's a broad-based response. In actuality, that increase is earmarked for certain things, right? And while vision research has gotten a portion of it, it's been a relatively smaller portion of that increase because, as you might imagine, it's getting poured into infectious disease and vaccinations, things like that. So that's good. But this day and age, it's always important to be diversified in how we support ourselves here at the university and in the Eye Institute so that we can keep the research going. So we have federal funding, which is a huge part of it. We have foundations, so there are different foundations that are interested in specific types of diseases like glaucoma, macular degeneration, retinitis pigmentosa, and our researchers are very effective at getting support from them because they like the research that we do. We also have company support, so people who want to develop clinical trials come to us and work with us to develop those clinical trials and to use the therapeutics that we're developing and patenting here at the University of Wisconsin. And then there's philanthropists, and so that's very important for us too. And that helps fund projects that are a little more aggressive and out there. So a lot of these other sources of funding, they're out there, but they want them to be safe. They want them to be things that they're pretty sure are going to work. What seed funding allows us to do in the Eye Institute, one of the things that we try and focus on is giving our researchers who are brilliant, but sometimes hamstrung by what they can do. You know, you'll get a grant, but that grant will say, you can do this, but I don't care what else you discover along the way, you can't color outside the lines. So a lot of the work that we do, or the funds that we get, say from Cycle for Sight, allow our researchers to color outside the lines a little bit, which then leads them to have the critical information to go apply for the federal grants and the millions of dollars to advance those projects. So those kind of pilot projects or seed funds are very important. So rest assured, we have lots of different ways that we keep things going. And since we're growing at such a rapid rate, it's very important for us to do that. And we watch those federal budgets very closely.
Okay, we've got a couple more questions. We have somebody who's called in on the phone.
Thank you. I have pseudoexfoliation syndrome, and it hasn't progressed yet to glaucoma, but I find in my searching that seems to be limited news on the subject. I wondered if maybe you have something to say.
Well, I always have something to say. Whether or not you're going to like it, I have no idea. So pseudoexfoliation. So now we're talking front of the eye. Again, I love all these questions because sometimes I get a one-track mind. But as I told you earlier, we're interested in everything from the front of the eye to the back of the brain in terms of our research here. So now we're talking about something that's in the front of the eye. So pseudoexfoliation is a condition where almost, and this is no reflection on you, but it's almost, think of it like dandruff inside the eye. Parts of the front of the eye release these tiny little bits of debris. And normally the fluid that's in the front part of the eye has to drain out what's called the trabecular meshwork. And so if you have these little bits of debris that your eye is making, they can clog it up, just like you clog a drain with hair. And so it's a special type of glaucoma. I mentioned before, there's many different types. So pseudoexfoliation glaucoma is a type of glaucoma. It's called a secondary open-angle glaucoma. So the approaches to that are first and foremost drugs to reduce the production of fluid so that it can reduce the pressure inside of the eye. And then there are surgical procedures that can bypass that drain altogether, to put a new drain into the eye to allow fluid to come out. So there are effective treatments, and most folks that have pseudoexfoliation glaucoma, if it does progress, have treatments that they can turn to that are effective in most of them.
Yeah, one problem in addition though is when you surgery such as to remove cataract, there's such a compromised sort of infrastructure as I understand it.
Yeah, so the lens itself in the eye, it has suspensory ligaments, and those can become very fragile. But let's say even if that were to happen, and if you were to have a complicated cataract surgery because those suspensory ligaments broke, there are other places that you can put an artificial lens that have been well worked out. They're called sulcus lenses. So in the vast majority of cases, even though people will say that there's a higher complication rate for cataract surgery in people with pseudoexfoliation glaucoma, and that's true, those complications in an experienced cataract surgeon's hands are very manageable. And most folks, even with those complications, have normal or near normal vision afterwards. So it's not something to be afraid of, something to acknowledge and to make sure that your cataract surgeon is comfortable working with patients that have that extra potential complication. But we're very, again, fortunate in Madison and at the University of Wisconsin, Davis Dewar, other places, to have very, very skilled cataract surgeons. So hopefully that takes away some of your anxiety about that.
I want to make sure we get to this question from Jennifer Shimon, which has been up here for a while. If there is a family history of RP and 2 children have night blindness, but they are now in their mid-40s and their vision has been stable to this point, is it possible they are safe from the progression to RP, or is progression possible at any age?
So for the most part, people with RP have onset such that in their late childhood, early teens, maybe early adulthood is when they first start noticing their symptoms. Now, I'll tell you, I'm a pediatric ophthalmologist myself, so I see a lot of these patients. And it happens so slowly that oftentimes they don't know that they have a problem years before it becomes apparent to them. So just because somebody recognizes that they have a problem when they're 30 or 40 doesn't mean that it hasn't been there slowly since they were in their 20s. And different types of retinitis pigmentosa have different progressions. So yes, most of them start early, some can start later, and there are very straightforward ways to diagnose this. So if there is a family history of retinitis pigmentosa and somebody has some night blindness at an early age, now you can have trouble with night vision just as a process of aging. So if you're in your 70s or 80s and you're not so good in the dark anymore, that doesn't mean you have retinitis pigmentosa. But if you have a family history of it, which is rare, only about 2 million people worldwide have retinitis pigmentosa, and you have a concern, regardless of your age, you can see a retina specialist or a person who specializes in ophthalmic genetics, and we have them here at the University of Wisconsin, Dr. Kim Stepien and Dr. Melanie Schmidt. And you can do relatively straightforward, simple tests that will rule that out or tell you whether or not you do or don't have it. That's a very good test that we have. So I encourage folks that have these concerns to just get it checked out. And then if it appears that they may have it, then the next step is to get genetic testing. And that's very important. Most folks, or about half of folks, they will find their genetic disorder if they have one on genetic testing, which is much better than it was many years ago. There are some gene defects that we still don't know very well, and we're working on trying to find all of them.
Okay, I see that Dr. Harry Roth's hand is up, and
He's probably going to correct me. Please.
that's right. That's what I thought. I wanted him to chime in.
Incidentally, I have pseudoexfoliative glaucoma that is doing very well. And one thing to know about that it's very unusually responsive to laser treatments if you do get the glaucoma. And the other thing is it's associated with elevated homocysteine levels in your blood. And some people recommend taking folic acid supplements for this. So these are things you can discuss with your internist and your ophthalmologist.
Thank you, Dr. Roth. I am still regretting that Dr. Roth has retired because he was my eye doctor for a little while.
I got to tell you, you can't get better than to have your question about pseudoexfoliation glaucoma answered by a glaucoma specialist who has pseudoexfoliation glaucoma. So that's about the pinnacle of advice right there.
You've got a question here from Joe and Blake. Dave, have you had success with your stem cell scaffold program in animal trials?
So the answer to that is yes. So one of the reasons why we've been able to advance that program so well is because we've now tested it in multiple different model systems. And it might come out as a press release, just like our Best disease work just came out as a press release by the hospital. We just earlier this year got a $6.2 million grant from the Department of Defense to use the patches that we make for soldiers who are injured with blast injuries, something called commotio retinae. So somebody who experiences a large blast and a concussive force to the face, that force is transmitted through the eye, much like if you got punched in the eye, and that can cause damage to the back of the eye through shock waves, and that causes photoreceptors to die. Or, unfortunately, we are seeing more and more laser injuries using military lasers and targeting lasers that are blinding soldiers. So we applied for, and I think it's only the second grant that the military has given out since it started giving out these grants about 10 years ago, to use our patches to hopefully be able to advance them and deploy them rapidly so that soldiers might be able to benefit from them as well. That's an example of how the same technology that could help retinitis pigmentosa or macular degeneration might very well also be able to help people who have direct injuries and young folks. Again, that's just one example of the Department of Defense. When it comes to medical research, they do their due diligence very carefully. And so we felt very honored that they looked upon our project. It's called the ORCA project. It's called the Outer Retinal Reconstruction for Combat Afflictions. And the nice thing is that the work that we're doing to advance that is directly going to advance its applications to all the other different things. So it's the same therapeutic. We're not changing it for retinitis pigmentosa or macular degeneration than we are for the soldiers. But they want to see, or they believe that it will be effective for these other types of injuries as well. So we're very pleased on how these are going, and we're in the process of working to try and get funding for these clinical trials, which cost, as I said, tens of millions of dollars. But we've been very fortunate to get a lot of folks that are enthusiastic about what we're doing here at UW and the IST.
Abe, I think you had an advanced question about the timeline for clinical trials, specifically with regard to ushers. And there's another question from Jim and Ellen Hunter, 201 again, as to whether COVID-19 has affected those timelines for clinical trials.
So this brings up a couple of good points. So I've talked in great detail at this point about replacement of lost photoreceptors in the eye. That's, of course, what my lab does. We're very near and dear to us. In terms of starting to see human applications, the helper cell, the retinal pigment epithelial cell, that's been in clinical trial now and in humans for over 4 years. Now that's a much easier cell to make, and it's not photoreceptors. It's not the cells that actually respond to light. So in terms of human applications for replacing photoreceptors, the hope there is that it will be within, again, the next 1 to 2 years. So this is where I get cornered a lot, and someone says, okay, when is this going to clinical trial? And the first thing I tell people is, I don't have full control over when that happens as a researcher, because I don't have a $10 million check I can write or a $30 million check that I can write. So I'm very careful to tell folks that everything is an estimate. I used to never tell people what I thought, how long it was going to take to get into a clinical trial, because I really didn't know. I've gotten a little more after, being in the game for a long time and kind of seeing how things are moving, a little more willing to kind of put my neck out there and say what I think it's going to be. So I will say that when the estimate that I gave was 2 to 5 years, and it's been over 2, so I'm still in that zone. So if this were Price is Right, I would still be in the game here. But I will give you an example of all the different forces that are in play when it comes to taking something, even that has tons of promise, to a clinical trial. So there's an example, and I can't tell you exactly what it is, of a therapeutic that was very close to combining with a big company that was more than willing and more than excited to put down the 30, 40, even $70 million needed to bring it to clinical trial. And everything was all right, wrapped up and almost ready to go. And then another bigger company bought that other company. And when that bigger company bought the company, he bought it for other reasons and basically says, we're not interested in the eye. And essentially kiboshed anything that they were doing in the eye. Okay, that's not anything anybody had any control over or could foresee. But it made you go back to the drawing board and go back out and say, okay, that company then had to go say, okay, who else is interested? And many other people were. But for an entire year, they were only negotiating with this one company because that was going to work out so perfect. So these are the sorts of forces that we all deal with. Some of them are on the research side, some of them are on the business side, some of them are on the COVID side. I mean, so COVID stopped every clinical trial for many, many months. I hate to say the word patients because people who have vision loss are told all the time, and they're probably getting sick of hearing it. But I think what we can do as researchers is make sure we keep the pedal down as much as we possibly can safely and go down as many avenues as we can and diversify the way in which we try and get these therapeutics to the patients so that we have all these shots on goal. So that's just one example. Now, I'll also say, once you get into humans, that's a phase 1 trial. Now, I won't get into all the details because this is a subject of a whole different Zoom meeting, but phase 1 trials generally have as little as 10 to 20 people in them. And they're designed to make sure that what you've just made is not harmful. And you have to watch those patients for at least 1 to 2 years. And if they're safe, then you can start bringing in more patients. And so it takes time. This is the process, and this is why you can't snap your fingers and have it done. And it's also why the researcher, or even the company that's licensing the research, doesn't always have the final say in when something actually gets to clinical trial.
I think that phase 2 and phase 3 would then get larger from there. I think
Much larger. So phase 3s can be hundreds to thousands of patients. The COVID vaccine that we all hear about, that Pfizer and Oxford Therapeutics are doing right now, are enrolling 30,000 patients in it. Well, that's different. It's 30,000 because we're talking about potentially vaccinating the world. Clearly, you can't do 30,000 retinitis pigmentosa people because that's a third of everybody with retinitis pigmentosa. So the numbers of patients are proportional to the number of people that are affected by the disease. And that's dictated by the FDA and other regulatory agents, not by the researcher, not by the company.
And you did touch on COVID-19 having delayed many or most clinical trials.
All of them, almost all of them, right? You're not allowed to be in person. So all in-person human clinical trials were put on hold, and that's not everywhere. But just last, like 2-3 weeks ago, the phase 2 reboot of the university began to reallow in-person clinical trials.
Dave, did you have anything you wanted to say before I wrapped up?
Oh heavens, I think I've said 2 hours worth of stuff. So I'll save something for a later Zoom, assuming that we didn't chase everybody else off.
We want to thank all of you diehards who are still on and have hung in, and everybody who's come on. This is really great for us to do, and we hope that you found it helpful as well. Please send us feedback, and please feel free to send me any questions if you didn't get a chance to ask a question that you had. If you're not on our mailing list to receive our newsletters, which we send out in the mail, then drop me an email and I can put you on that. We don't inundate you with paperwork, but we send them out 3 times annually, as well as a year-end update. And finally, I just want to thank all of you for all the support that you have given us. All of what we do here is in large part due to your helping out, and we really appreciate it. So have a great night, and we'll do this again.
Yep, thank you. And please give us your advice in terms of how you might like to see things done differently. For instance, we could have disease-specific ones. We could have smaller ones where we focus on AMD or glaucoma or the front of the eye or the back of the eye. That might be helpful too. So all those sorts of things I think are important. But as a first attempt at this, we really appreciate you guys beta testing it for us. And we look forward to getting your feedback. Most importantly, we're grateful to everybody having faith in us and supporting us. And so this is a way for us to give back, and it's important, and it also kind of energizes the research that we do and helps to focus us. So I'm going to say thank you as well, and I hope we have a good rest of the week and a great weekend.